Thank you. Good afternoon, everybody. It's a really good crowd considering the number of people I saw at the bar on the way over here. It's great to be here again and update you on the progress we've made at Homology Medicines over the last year to 2 years now. Forward-looking statements, I'm glad we can skip by that slide. It's not Reg FD. When I come to the Mesa, I really see the crystallization of genetic medicines and the ability now to actually talk about curing diseases versus a world that we've lived in for a long time where you can only treat a disease. So this is what really drives us and everybody here in the room at Homology in terms of being able to treat and potentially cure patients living with rare genetic disorders. So Homology is a fully integrated company with a dual platform for gene therapy and in vivo gene editing. Technology really involves 15 novel AAV capsids that were isolated from human stem cells. And as an immunologist, I really like that profile because it would lead you to believe that there's a safer immunogenicity profile, which looks to be true in our case so far. It's a leadership team with a lot of rare disease expertise. Together, we've launched 11 different rare disease drugs helping patients today. We really have a very good engine. We've had 5 development candidates in the last 5 years, led by my colleague here, Albert Seymour. And that's really led us to the point where we plan to have 3 clinical trials by the end of this year. And we just announced today the commencement of our gene editing trial for PKU earlier in the afternoon. We are in a phase 2 for our gene therapy trial for adult population of PKU, so that's exciting. And then I'd like to highlight our internal manufacturing expertise. Everybody, I can't understate enough how important it is to have control over your destiny on that one. So these 15 AAVs belong to the same family, and they differ in just a few amino acids, but those few amino acids translate into very different biodistribution profiles. And it really underpins 3 approaches here: gene therapy, what we're calling our GTXMap, or ability to make fully formed IgG, heavy and light chain IgG, and then also our in vivo nuclease-free gene editing. That means it's not CRISPR. And I also like to highlight that we have published now biodistribution from almost all of these capsids and shown very different biodistribution patterns. And we have a couple of vectors that actually detarget the liver and go directly to skeletal and cardiac muscle. So a very exciting platform from which to work. You can see that off the same capsid, we can drive 3 different parts of this platform. You can add a gene through gene therapy. You can make a monoclonal antibody off of our GTXMap platform. And then you can also permanently correct the chromosome and do gene correction with our in vivo gene editing by homologous recombination, which is a very precise DNA repair mechanism. Here's a look at our pipeline. Continues to move to the right, which is always a good sign in these 3 areas. And really, I'll focus now on the PKU franchise, where this disease really remains at a very high unmet medical need. On the right, you see the pathway here. Essentially, these patients are identified at birth and fail to make PAH, which is a naturally occurring enzyme that converts phenylalanine to tyrosine, and tyrosine is an important precursor of all the neurotransmitters. So the buildup of phenylalanine is neurotoxic, causes problems for these kids. So right out of the hospital, these kids begin to lose executive function, and they have to go on a protein-free diet. 95% of the patients out there have no real therapeutic, and standard of care is a highly restrictive diet, which is very difficult to follow. It's a very large rare disease population. There's about 50,000 patients worldwide. About 2/3 of these are adults. But also every year you have about 1,000 to 1,500 babies born with this disease. And so this is a recurring population that's very large. So our approach to do gene therapy in adults really is meant to address the adult population with PKU. And our approach with gene editing for the pediatric population of PKU is meant to move towards the ped population so that these kids can be treated at a very young age and not even remember that they were diagnosed with PKU. That's the goal here. So our first trial is called the FIXX trial. That's HMI 102. We're in a phase 2 dose expansion phase right now. In the phase 1, we showed the dose escalation trial a great safety profile In addition, we were able to select 2 doses to move forward into the dose expansion phase. We did a lot of learning along the way there and optimized the protocol for our dose expansion phase, which is ongoing right now and enrolling. We expect to report out in mid-2022 on an update on that data set. We have a total of 13 clinical sites, and we're adding more as we get to the end of the year. And we're very, very excited about the progress here. We've seen good data that prompted an investment from Pfizer when we released a data set at the end of November last year. This is a trial design. It's a 2-to-2-to-1 randomized control trial. We selected a dose of 6E13 as well as 8E13, and obviously running a control, which will be crossed over to one of the treatment arms at 26 weeks. So this trial is ongoing as we speak, and so very excited about the data we've seen so far and looking forward to the future. We announced earlier today that we will begin our FIXX trial for HMI-103. This is our nuclease-free gene editing candidate for the pediatric population of PKU. We'll start in adults and eventually move to a younger population. It uses the same vector or capsid that we use for HMI-102, which is AAV-HSC15. So we have a lot of safety data around the HMI-102 program that we can apply to this trial. We've already have been engaging with multiple clinical sites, and we plan to get this trial started by the end of this year, and just received FDA clearance very swiftly, I would add. This is the preclinical data showing how just one IV administration of HMI-103 reduces phenylalanine down to wild-type normal levels. What's nice about this approach is it uses homology arms to direct the PAH gene to the exact position in the chromosome where you want to make that correction. And you can track this very readily, so you can very easily detect on-target and off-target mutations that could potentially occur. And so what you really want to do here is to identify, and this is what FDA is very interested in right now, is where is this big stretch of DNA going, and is it on target or is it off target? And because it's homologous recombination, we have a highly specific integration efficiency with little to no off target within the area of the assay. So very excited about this program moving forward. Talk to you briefly about 2 other programs we have. Our MPS II Hunter program, which is a monogenic lysosomal storage disorder that you heard a little bit about in the previous talk, and also focused on metachromatic leukodystrophy, also a lysosomal storage disorder. These are both diseases that have a peripheral nervous system manifestation and a central nervous system manifestation. Jeff, I'm saving you a couple of slides on your talk with the Hunter program. So we are planning to initiate our trial for Hunter this year. That's on track. And metachromatic leukodystrophy remains an optimization of the vector phase, so it's still in preclinical and moving along. Here you can really see the manifestation of the peripheral disease, and these are patients who are currently mostly on enzyme replacement therapy. A lot of us had a lot to do with that, the launch of Elaprase back in 2006, 2007. We will focus on the peripheral form of the disease. There's still a very high unmet medical need there. These patients still don't live past the age of 45. They still have cardiac problems, still have lung function, still have joint pain. So we'll target there first and target a 6-minute walk test as an approvable endpoint there. And then the learnings there will be then able to allow us to move to a younger pediatric population where we can track the reduction of GAGs in the CSF and the crossing of the blood-brain barrier by these vectors, which they do very well in non-human primate models. So we're very excited about getting this trial off the ground at the end of this year, and we're on track for that, so stay tuned for more news there. These are the preclinical data that went into the IND submission. It shows both biochemical correction of the disease, reduction in substrate, as well as phenotypic correction and functional correction. So those are important markers when you're looking for an IND clearance. And we show sustained levels of I2S, not the peaks and troughs that you would see with an enzyme replacement therapy. We significantly reduce GAGs here, which is a substrate that builds up. And we have phenotypic correction of joints and skeletal features in the animal model of this disease. A little bit of preclinical data on metachromatic leukodystrophy. What we did on the left-hand side I just alluded to the fact that these vectors cross the blood-brain barrier very nicely after IV administration. On the left, you can see that you get very good biodistribution in the brain of a mouse here compared to AAV9. So we see it in all regions of the brain. We've also replicated this in non-human primates, which we all know is really the gold standard for assessing whether or not something crosses the blood-brain barrier. And then we've shown the functional benefit on the right-hand side with correction in the rotarod. These mice are able to navigate now after a single IV administration of our MLD vector. Lastly, to talk about our GTXMap platform. Again, here, it's the ability to constitutively produce IgG from the liver. And this is a full heavy and light chain IgG. We've selected C5 as the first target. Obviously, this is a target that's well known with the development of Solaris and Ultomiris. And it's a target that can be knocked down very hard without any real safety issues. So we're really excited about this program. This is essentially allowing us to use the liver as an antibody factory so that you can see constitutive normal expression of the monoclonal antibody or the anti-C5. And you can see the preclinical data here. This preclinical data allowed us to select a clinical development candidate actually ahead of schedule. And so we're in IND-enabling studies with this drug now. And as you know, C5 also is a key mediator in diseases like myasthenia gravis and aHUS as well as a number of other diseases. So a lot of opportunity to grow there as well. Finally, last slide. Manufacturing has been a priority of ours from the very beginning. We invested heavily in the early part of the company in process development, early vector design, and optimization, and have grown. It's more than half of the company now that does our own internal manufacturing. On site, we have a 25,000 square foot facility with 3 by 500 liter. We also have scaled up to 2,000 liter, and we have that capability. We're at commercial process and commercial scale, and the quality of our manufacturing material is very high. And I can tell you, having gone through a summer of 2 INDs going through FDA, you better have very good analytics and very good quality AAV if you want to start an AAV trial these days. The bar is much higher than it was in the past. So we're very proud of our manufacturing prowess. It's never been on the critical path for us in any of our timelines, and it was a great investment. Even my CFO would agree to that. So here's our pipeline now. The company is 5.5 years old now. Very proud of the execution we've been able to deliver on behalf of our patients, and we're looking forward to a number of milestones toward the end of this year and into next year. And with that, I will end. We have a couple of minutes for questions if anybody wants to fire away, but thank you for your attention.